Welcome to the very last PowerPoint video about macromolecules in living things. If you've watched the previous videos, we've talked about carbohydrates and proteins and lipids. And this very last video now is going to be about our last macromolecule, which is nucleic acids. Nucleic acids contain carbon and hydrogen and oxygen, just like carbohydrates do, just like proteins do. The atoms, in addition to those basic three that are found in nucleic acids, include phosphorus and nitrogen. Remember, we saw nitrogen also in our protein molecules. Nucleic acids are built of this little subunit. It's called a nucleotide. Look at the beginning. Nucleotides, nucleic acids. These are the building blocks. The little pieces were joined together to make a nucleic acid. Nucleotides have this basic shape. They have a sugar molecule attached to a nitrogen base. And remember P for phosphorus. This is a phosphate group that has a phosphorus atom in it. And depending on what kind of nucleic acid I make, I'm going to change the nucleotide subunit a little bit. And the two things that I can change are the sugar that I put in the sugar spot and the nitrogen base that goes in that nitrogen base spot. All of those different subunits that I make that are nucleotides have different sugars and different nitrogen bases to make them each a little bit different. So if I'm going to use that basic nucleotide shape, in the sugar spot, my choices that I have for putting in that spot are this sugar. It's called deoxyribose. And if I'm going to make a DNA molecule, that's the sugar that's going to go into that sugar spot. If I want to make an RNA molecule, I'm going to use a different sugar. I'm going to use ribose sugar. Look at the two molecules. The difference between these is this oxygen is missing. In a deoxyribose, this oxygen is been removed. Deoxy, lose the oxy. Deoxyribose is ribose sugar that has lost an oxygen. Those two kinds of sugar, and they're very similar in structure, are used to make the basic building blocks to build a nucleic acid. So depending on what kind of NA, what kind of nucleic acid I want to build, I'm going to put one of those choices into the sugar spot. For nitrogen base, I have also some choices that go in that nitrogen base spot. I can use adenine, and the abbreviation, the shorthand for talking about adenine is use the letter A, the first letter of the name. G for guanine, C for cytosine, T for thymine, U for uracil. Notice all of those end in ene, I-N-E, except the uracil. And notice also both DNA and RNA contain these A's, G's, and C's, these nitrogen bases, but thymine, the T one, is only found in DNA, and uracil, the U, is only found in RNA. So if I want to take that little basic subunit and I want to make a DNA molecule, I have choices. I'm going to use either an A, adenine, a thymine, T, a cytosine, C, or a guanine, G, in the nitrogen base spot. And notice no uracil. Remember, uracil is only found in RNA. So if I'm making DNA, I can't use uracil as one of my choices for a nitrogen base. In the sugar spot, I'm going to put that deoxyribose sugar. And so once I put my nucleotide together, I can use that to join together to make a bigger molecule, a nucleic acid. If I'm going to make RNA choices for nitrogen bases, I can use A's or U's or C's or G's. Remember, if I'm making RNA, can't use the T, no thymine in RNA. The sugar that goes in the sugar spot for making RNA is ribose sugar. And now that I have put my nucleotide together with a ribose sugar in one of those nitrogen-based choices, I have a little subunit I can use to join together to make a bigger molecule. Here are those nucleotides. And if I want to join little pieces together, think about what we talked about. Remember in our other slideshows when we talked about joining molecules together, and we kind of compared it to building a pot bead chain. Each of the little pieces can join together to make a bigger chain and make a bigger molecule. And we use this reaction to take glucose, and make a polysaccharide. We use the little pieces, amino acid pieces, amino acid, amino acid, amino acid, to make a chain of amino acids. That's a protein. And we're going to do the same thing. Now we're going to use nucleotide little pieces, subunits, to go together, nucleotide, 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 to go together to make a nucleic acid. So remember our reaction, the chemical reaction we used to join little pieces. Pull off the atoms, a little from each side, hydrogens and oxygens to make a water molecule, and join the remaining parts together to make a bond. Remove water, join, and make a bigger molecule. Dehydration, that's the remove water part. Synthesis means I'm taking little subunits, little monomer pieces, and making a bigger molecule, a polymer. DNA is a polymer made by joining nucleotides. If you look at the DNA molecule, DNA, deoxyribonucleic acid, is a nucleic acid, Na, that has deoxyribose sugar, DNA. It's double-stranded. There's two strands, one that runs this way, one that runs this way. They go together, and notice those nitrogen bases, the A's and C's and G's, form the rungs. It looks kind of like a ladder, the rungs of the ladder. And the subunits that I can use, remember, to make make DNA are those A's, T's, G's, and C's. No U's. If I want to make that combination now, I put my ladder together, and DNA is what we call a helix. It looks like the ladder's been twisted. If I look at how those little units go together, always those nitrogen bases run across the middle between the two sides of the ladder. They form the rungs of the ladder. And when they join up, the adenine, the A's, always bond across the ladder. They hold hands across the ladder with thymine. So if I look at this molecule and I see an adenine here, I know that this one across the, holding hands across the ladder, is got to be A's go with T's. It's got to be a thymine. And when you look at the ladder and how it goes together, the C's, the cytosines, always bond with a guanine. So if I see a cytosine on this side, I know this one that's holding hands across the ladder has to be a G. C's go with G's, A's go with T's. RNA, ribonucleic acid, it's a nucleic acid, an Na that's got ribose sugar, RNA, is single-stranded. It's one side of that ladder. Notice the nitrogen bases still stick out towards the side, but there's not a match, another side to the ladder. RNA is single-stranded, and the bases, the nitrogen bases I can use to make RNA are A's and U's and G's and C. Remember, no T in RNA. What do nucleic acids do for you? First of all, those little building blocks that go together to make a nucleic acid, we use one of those nucleotides to be our energy storage molecule, store and transport energy. We talked about this molecule back when we talked about doing dehydration synthesis and hydrolysis. Lose water to make a bond, add the water back to break a bond and release the energy. So if I look at this little subunit, ribose for my sugar, the A choice, adenine for my nitrogen base, If I just looked at this part of the molecule here, the part that has that little nucleotide subunit, here's my sugar, here's my nitrogen base and phosphate group. That little piece I could use as a subunit with ribose here to make an RNA. But if I add more phosphates, I make a 3-phosphate, there's those P's, 3-phosphate molecule. And remember, this is an energy bond. If I add the P here, it stores energy, and breaking that bond to release the P, releases energy. This molecule, it's got ribose for the sugar, it's got adenine in the nitrogen base spot, and it's got three phosphates. This is called ATP. ATP. The TP, T stands for tri. Think triangle, tricycle. Tri means 3. And the P is for the phosphate. It's got 1, 2, 3 phosphate groups attached to it. And it acts like a rechargeable cell phone battery. Remember, we talked about how we can charge up this molecule by adding the phosphates and then release the energy where I need it to do something in the cell by removing the phosphates. And we talked about how dehydration, synthesis, and hydrolysis can work together to charge up the P's and remove those P's to be our energy storage molecule. If I take lots of little subunits and hook them together to make a big DNA molecule, the function of DNA in a cell is to store genetic information. It's our gene molecule. It makes the genes. The sequence of those A's and T's and G's and C's are what determine how cells are going to act. It carries the information to tell the cell what to do and how to be. RNA also is an information molecule. It's going to carry the information from the DNA in the nucleus out to the cell. It's the connection between the information that's stored in the genes and carries that information out to the cell to tell the cell what to do. Also, RNA helps with protein synthesis. Remember, synthesis means to make or build. So one of the ways that the information moves from the DNA out to the cell is by telling the cell how to make a protein that's going to do something in the cell. And RNA helps with that transfer of information from the DNA out to the cell to make proteins and allow the cell to do things. That's the very last thing we need to have a minute to think about, is that connection between shapes of molecules and what they have to do with how that molecule does its job. Shape of a molecule is pretty important. Think about shapes of swim flippers and how they would help you to move through the water because of their shape. Shape of a hammer is important for pounding a nail. And molecules all have shapes. Remember we talked about 3D shapes, folding of proteins, the shapes of carbs, the shapes of DNA molecules. All of those molecules have different shapes, and it's because they do different jobs in cells. I want you to take a minute and think about those molecules that we learned about and what their shape has to do with the jobs that they do. That's the end of our slideshow talking about molecules, and thanks for watching.